This meeting is being recorded.
Okay. It's 12:01. I guess we can start. So thank you everyone to join the ARO seminar series. Today we're going to start the topic of auditory brainstem and midbrain. We're going to kick off the series with Dr. Paul Manis as our speaker today. My name is Ray-Li Xie. I'm an associate professor at the Ohio State University. I will be the moderator for today's webinar. So before starting today, if you have any questions during the presentation, please put the question in the chat so we don't interrupt the presentation. After the talk, you can ask questions by raising your hand and unmute yourself to direct ask. Or you can also put questions in the chat, and if we don't have enough time, Dr. Manis will answer them by email. So closed captioning for this meeting is available, and this session is being recorded and will be made available for additional viewing. First of all, Dr. Paul Manis, he's currently the Thomas Park Distinguished Research Professor in the Department of Otolaryngology, Head and Neck Surgery at the University of North Carolina at Chapel Hill. So he received his bachelor's degree from California Institute of Technology, and then PhD in neuroscience from University of Florida. And he went on for a postdoc training at Vanderbilt, and then started his faculty position in 1985 at Johns Hopkins University. In 1999, he moved to UNC Chapel Hill and stayed until today. As we all know, that his primary research is the cellular mechanisms of auditory information processing in the cochlear nucleus. And the work including mostly synapse transmission circuits, ion channel function, and development of biophysically based models representing neurons and circuits of the cochlear nucleus. And also he has other projects working on auditory and prefrontal cortex. So I actually got my postdoc training in Manis Lab from 2007 and 2014. I'm really honored to learn basically everything I'm doing in my lab from Manis Lab by studying the mechanism hearing loss in the cochlear nucleus. So I also want to make that Dr. Manis served on multiple NIH grant review committees, the NIDCD Council, Board of Scientific Counselors, and also the chair of the Blue Ribbon Panel on the NIDCD intramural program. He has been the principal investigator on multiple research and training grants from NIH. And his talk today is The Cochlear Nucleus, a Perspective. Without further ado, it's my pleasure to welcome Dr. Paul Manis. Thank you.
Thank you, Ray. And I'd like to thank the organizing committee for inviting me for this talk. And so, yeah, I'm going to talk about the cochlear nucleus in a perspective. I don't have any conflicts of interest to declare. The work I'm going to be talking about is supported by several NIH grants here, one of which is actually mine, and the other two, the ARO one is with George Spiru. Actually, I won't be talking about that work. And an RF one that I have with Hiroyuki Kato, which is a Brain Initiative grant. We'll be talking about that one. And then I'm going to be talking about some work from past grants that we've had, including all the way back to an NSF grant that I had as my first grant as my first federal grant as a investigator. Okay, so this is an outline of the talk. I'm going to talk about the cochlear nucleus and give you an introduction to it. A brief introduction is a system of intertwined and parallel pathways. Talk a little bit about serendipity in science and something that we did looking at the intrinsic discharge patterns of DCN pyramidal cells. It's kind of something old, an old story. And I'm going to talk about work that we're currently doing, looking at hearing loss, the effects on intrinsic excitability of neurons in the dorsal cochlear nucleus specifically. And then we're going to take the long view, what I call the view from the top, and look at a short latency pathway from the cochlear nucleus to layer 6 of the auditory cortex. This is collaborative work with Hiroyuki Kato's lab. And then I have some closing comments. Oh, wrong button. Okay, so this is a view of the cochlear nucleus in a parasagittal section. This is kind of the easiest way to look at the layout of the nucleus. The nucleus consists of 3 broad parts: an anteroventral cochlear nucleus, a posterior ventral cochlear nucleus, divided by the entry region of the auditory nerve, and then the dorsal cochlear nucleus, which sits on top of the rest of the nucleus. The anterior part of the brain, or towards the front of the brain, is on the right, towards the back of the brain is on the left. And the auditory nerve enters ventrally and laterally into the nucleus. The fibers then bifurcate into an ascending branch that goes into the anteroventral cochlear nucleus and a descending branch that goes into the posterior ventral cochlear nucleus. And they bifurcate in a tonotopic arrangement such that fibers from the most apical region of the cochlea bifurcate first, and those from the more basal regions representing high frequencies bifurcate later, which helps form a tonotopic map of across the structure of the nucleus, going from low frequencies to high frequencies here in the ventral cochlear nucleus, in the anterior part, low frequencies to high frequencies in the posterior ventral part of the nucleus. And then there's a flip or a change in the map, and the dorsal cochlear nucleus then goes from low frequencies to high frequencies, following the same kind of axis as you go dorsally. The fibers, as they bifurcate, the posterior ventral cochlear nucleus fibers go through a nexus, perforate this granule cell domain that divides part of the dorsal and the ventral cochlear nucleus through little perforations or lacuna in the granule cell domain before it goes up into the dorsal cochlear nucleus to form similar lamina. The different principal neurons of the cochlear nucleus are indicated here, and a little bit about the functions that we sort of canonically think that they're involved in, starting with the bushy cells, which are amongst the best studied cells in the nucleus. They receive input from the auditory nerve with large endings called end bulbs of Held. They project into the superior alveolar complex, and they're really good at looking at conveying information about the timing and fine temporal structure of sounds, such as formants and cues that are used for sound localization for low frequency sounds. There are a fairly large population of cells in the anterior ventral part of the nucleus. Scattered both in the anterior and posterior parts of the nucleus, at least in the mouse, are the planar multipolar cells, or we refer to them as T stellate cells. Every cell in the nucleus seems to have multiple names just to confuse people, so we'll call these T stellate cells for most of this talk. These convey information about intensity of sound and the envelope of sound, sort of low frequency fluctuations in the amplitudes of sounds. And they're not so good at conveying the fine temporal structure of sound. They project both within the nucleus, as I'll show, and also mostly to the contralateral inferior colliculus. In the very caudal part of the nucleus, nestled up against the corner of the granule cell domain here, are the octopus cells, a modest population of cells which have some really unique features and are some of the best timing cells in the nervous system. And they convey information about timing and pitch, which goes mostly into the superior alveolar complex. Another set of cells in the cochlear nucleus that are projection neurons are called the radiate multipolar cells, or the D stellate cells, because they project dorsally. I should say the T stellate cells, the name comes because they project out the trapezoid body. These cells are a multipolar cell that projects dorsally out the acoustic stria, the dorsal acoustic stria. They have large radiating dendrites which cross many isofrequency sheets. They have broad receptive fields, and they project as an interneuron, because they're an inhibitory interneuron, they project within the cochlear nucleus to all regions, as I'll show in a second, and they also project to the contralateral cochlear nucleus. So they're a projection neuron. They're kind of unique in that respect. And finally, in the dorsal cochlear nucleus, the major set of projection neurons are called pyramidal cells or fusiform cells. And I will use both terms somewhat interchangeably here. And they convey information about spectrum as well as multisensory relationships of sound. And they project to the contralateral inferior colliculus with maybe only a few collaterals within the cochlear nucleus. The nucleus also is a host to a variety of inhibitory interneurons, a couple of classes of which are shown here. So I talked about the radiate multipolar cells, but now we're going to show the axonal projections of these cells. They project to the bushy cells, to the planar or multipolar cells, or the T stellate cells, and they also project up into the dorsal cochlear nucleus, where they play a very important role in shaping the response areas of the tuberculoventral neurons and the pyramidal cells. Tuberculoventral neurons are also a glycinergic interneuron that have local projections restricted to the cochlear nucleus. They project to the pyramidal cells, to each other, forming a interleaved circuit, and they also project to the ventral cochlear nucleus, where they end on most of the major cell types within the ventral cochlear nucleus. They're called tuberculoventral neurons because the dorsal part of the nucleus used to be called the acousticum tuberculum, and as its name, tuberculoventral, was given by the early anatomists because they project to the ventral cochlear nucleus. On the right, we have a picture, of actually a photograph from Kirsten Osen of a silver-stained cochlear nucleus taken in an axis that's kind of at an odd axis. It's at an angle, but parallel to the way that the tuberculoventral tract runs. And this shows the dorsal cochlear nucleus caudally. Here's the auditory nerve down in the back corner here. There's a set of fibers running in here, which are probably descending inputs to the nucleus. Here's the molecular layer, the DCN, unmyelinated region. Here's the anteroventral cochlear nucleus, where you can see cross-cut fascicles of auditory nerve fibers. And in between these, you can see this band of myelinated fibers going between the dorsal and the ventral cochlear nucleus. And this is what's called the tuberculoventral tract, which has axons of the T stellate cells, of the tuberculoventral neurons, and probably the radiate multipolar or D stellate cells. So there's a big connection between the nuclei here. So the nucleus is really integrated. If you think about the response properties of the cells, the excitatory cells, they are bound together in some respects by these interneuronal circuits, which they share. Finally, another projection here that I just briefly mentioned is from the T stellate cells or the planar multipolar cells. They also project into the dorsal cochlear nucleus, where they have excitatory collaterals whose function in the DCN is not really fully understood. Now, the connections between the dorsal and the ventral cochlear nucleus are tonotopic. To illustrate this is a study that was done by Luc Campagnola in my lab several years ago. He took brain slices of the nucleus and he cut them parallel, in this case, to the tuberculoventral tract, and he used glutamate uncaging to excite neurons in different regions of the nucleus. So this is a method where you use a caged form of glutamate that is inert. When you flash an ultraviolet light on it, the cage is broken. You get, within microseconds, you get glutamate at a high concentration right in the region where that flash was, and it can excite the AMPA receptors and NMDA receptors in the cells that are in that region and can drive them to spike. So we use a laser scanning method to do the glutamate uncaging, and mapped inputs to neurons both in the ventral, mapped inputs to neurons in the ventral cochlear nucleus. On the left, we show a bushy cell, which is located down here, and responses to stimulation show, for example, in the dorsal cochlear nucleus, this very small, narrow region produced inhibitory synaptic potentials. So these are outward currents that are generated from the uncaging. And these are kind of scattered in time because the uncaging excites the cells and causes a burst of action potentials in the presynaptic cells. There's also local inhibition that's seen around the region of this bushy cell. Likewise, recording from stellate cells, here's a filled stellate cell and its location in the ventral cochlear nucleus. Locally, there's a large amount of inhibition over a wide region that converges onto this cell. And in the dorsal cochlear nucleus, there's this very narrow band of inhibition that can be generated from the uncaging. And if you map from, if you compare the locations of the inhibitory inputs from the DCN to the locations of the cells in the VCN, what you see is a regular arrangement where the ventral cells in the antroventral cochlear nucleus and the ventral cells in the dorsal cochlear nucleus are connected together, whereas cells that are dorsal in the high frequency region of the ventral cochlear nucleus are inhibited by sites that are in the high frequency region of the dorsal cochlear nucleus, corresponding to this tonotopic map. And if you just simply map the locations against each other, you get a fairly nice linear relationship for both the bushy and the stellate cells. So this mapping of the interneuronal connections, at least from the DCN to the VCN, is also tonotopic. So when we look at these circuits, just schematically, you can think of the ventral cochlear nucleus as having this particular organization. We have excitation that's tuned to a particular frequency of auditory nerve fibers corresponding to the isofrequency sheets that ends on bushy cells and planar multipolar or T stellate cells. And those cells also receive input from other cells. So the vertical cells provide a narrow band inhibition input that goes through the dorsal cochlear nucleus, but they're excited by the auditory nerve, and it's basically on CF. We also have off CF excitation as well as on CF excitation from the auditory nerve to the radiate multipolar cells, the wideband inhibitors, which then also project to the bushy cells and the planar multipolar cells, and they also project, importantly, to the vertical cells in the DCN. This terminology and portions of this circuit were actually developed in studies of the DCN and work that was done in Eric Young's lab with Elian Elkin and George Spiro and many others. There's another set of inhibitory interneurons in the VCN that I didn't talk about. These are called L-stellate cells because they're local stellate cells. They're small multipolar neurons, and they also provide inhibition, and their function within the circuit is really not fully understood. What about the circuit in the dorsal cochlear nucleus? Well, it's kind of the same. Again, because these interneurons are shared across regions, we have the excitatory input to the pyramidal cells, excitatory input to the vertical cells, and on CF arrangement of, so the same frequency of inhibition, vertical cells get the same input frequency and provide inhibition at that frequency to the pyramidal cells. That's your narrowband inhibitor. And we have off and on CF excitation from the auditory nerve fibers through the radiate multipolar cells, providing wideband inhibition to these cell types. There's no L-stellate cells, but the cartwheel cells, which are another kind of interneuron enmeshed in the circuitry of the molecular layer of the dorsal cochlear nucleus. The cartwheel cells are akin to Purkinje cells in many ways, derived from the rhombencephalic lip and a portion of the cerebellum that gives rise to a portion of the developing brain that gives rise to the cerebellum, but they also give rise to these portion of the dorsal cochlear nucleus. These cells receive, amongst other things, multisensory input, for example, somatosensory input, possibly vestibular input, as well as many descending connections from different regions of the brainstem, including non-auditory as well as auditory. And these also provide inhibition. So like the L-stellate cells in the VCN, there's another source of inhibition in the DCN. So the circuits are similar. What differs is in the relative strength of inhibition between these different components and the relative strengths of excitation, which then help generate different kinds of response properties for these cells. So I just wanted to also mention that the time course of inhibition is specific to the target cell types. And this was a little bit of a surprise. Whoops. Okay, move the mouse and things change. This was a little bit of a surprise to us. And sort of a serendipitous discovery after many years of people looking at the cochlear nucleus. The inhibition was, I think, generally assumed to be very similar in all regions. But in these experiments done by Rayleigh in my lab several years ago, he took a brain slice, parasagittal brain slice, stimulated in the dorsal cochlear nucleus, cut in some experiments the auditory nerve fibers as they ran up into the dorsal cochlear nucleus, basically at the top of the PVCN here, and then recorded from bushy and stellate cells in the ventral cochlear nucleus. This is the slice that you can see in the chamber with a cut. Here's the ventral cochlear nucleus, auditory nerve root. Here's the dorsal cochlear nucleus. I'm sorry, auditory nerve roots here. Here's the anterior ventral, posterior ventral, and dorsal cochlear nucleus. When You record from these cells and you inject current. You get the typical response of a bushy cell consisting of one or a few spikes. The membrane potential is then clamped because of the activation of a low voltage activated potassium conductance. No more spikes are generated in response to a steady current step. Hyperpolarization gives rise to this typical SAG response, which is mediated by hyperpolarization activated nonspecific cation channels. So this is typical for bushy cells. The stellate cells have a regular discharge characteristic when you inject positive current here. They fire regularly. When you hyperpolarize, they show a similar kind of sag and occasionally an offset response with a few spikes. When you then go to voltage clamp and you stimulate the dorsal-cochlear nucleus, you can get an inhibitory synaptic potential, and this is flipped in sign because it's recorded in a high chloride solution, but inhibitory synaptic potential that in the bushy cells is actually relatively slow. The decay time constant here is about 9 to 10 milliseconds. Whereas in the stellate cells, and remember, these cells are the ones we think of as being involved in the fast timing circuitry, but they have this slow inhibitory potential. And the stellate cells, in contrast, have a very fast decay time constant of about 1.3 milliseconds here, very close to the time constant of their auditory nerve input decay. This is true for the evoked responses, and it also is true for the spontaneous synaptic events that occur in the slice from the spontaneous release of vesicles from these terminals. And in both cases, the responses are completely blocked by strychnine, so they're glycinergic synaptic input. And if you look at the iPSCs, spontaneous events, they have a very similar separation of fast decay in the stellate cells and slow decay in the bushy cells. And this also shows up even if you plot the rise time against the decay time for all these. They're categorically different in both classes of cells. This actually has important consequences for things like detecting fluctuating tones in the presence of fluctuating maskers, such as in the process of comodulation masking release. We're having a really fast decay time constant of inhibition turns out to be really important in improving the detection of tones during the quote-unquote dips in the sound in the comodulation masking, one of the comodulation masking release paradigms. And there's more detail about that in this paper. The other way the cells differ is in the temporal patterns of response and the frequency response areas. So the traditional way that cells were initially characterized in response to short tone pips was by their post-stimulus time histograms. These are schematic representations of those. So the bushy cells, both classes of bushy cells, and I'm not going to differentiate them any further here, but they both have primary-like or primary-like with notch kinds of responses. These cells fire irregularly, and so in response to a tone, because their inputs are irregular, and so the timing of the spikes on the output being driven by these inputs is also irregular, and over many repeated stimuli, it kind of averages out to look like this. The T-stellate cells have what are called chopper-type responses. So they have a regular discharge pattern, and that pattern is fairly repeatable from one trial to another. They accumulate a little desynchrony over time, so you lose that. But you can see at the very beginning of the response that the responses have these modes of firing, which give rise to the chopper name and are due in part to the regularity of their firing in response to depolarization. The octopus cells are known as onset cells. They can have onsets with or without some activity after them in response to a tone, although they can respond to modulated stimuli up to very high frequencies, up to 1 kilohertz. And onset choppers are D-stellate cells. They have this chopping response primarily at the onset, and then so irregular activity after that. In the dorsal-cochlear nucleus, we get 2 additional classes that were originally described by out of the group at Mass Eye and Ear, Nelson Kiang, Don Godfrey, Russ Pfeiffer. These are the pauser response, which shows an onset of the tone burst, shows an early high synchronous firing, followed by a pause in the response before a resumption of firing. and a buildup response, which is the same as the pauser, it just lacks this onset response. And these cells also show chopper type responses. The other categorization that's been used is the frequency response area maps. And just for illustration here, this is the frequency response area map for an auditory nerve fiber tuned to 4 kilohertz. All of the response is excitatory for combinations of frequencies and intensities within this black area. Outside of that, there's just spontaneous activity. And if you go up at the characteristic frequency here at this white dashed line, the response increases over some range. And as we know, different kinds of auditory nerve fibers can vary in their spontaneous rate and the intensity range over which they respond before saturating. So these don't show any inhibition. In the cochlear nucleus, though, inhibition becomes really important. And particularly in the dorsocochlear nucleus, there's another type of response. It says a name, it's called the type 4 or sometimes type 5 response area, depending upon the structure, that's seen in the fusiform cells and in the giant cells. There, the cells can have spontaneous activity. They have a low threshold area of excitation, but as you go up in intensity, they become inhibited. And that inhibition is provided by the 2 inhibitory inputs, the tuberculoventral cells and the D stellate cells. And
I already
most
add
of the
less
response, yes,
buffer.
most
I
of the
don't know if Caleb was
response,
that small.
I hear a voice, is most of the response area is inhibitory, and the excitatory areas, you get additional excitatory areas at other frequencies and intensities, which are kind of idiosyncratic to the cells. And if you go up in intensity, you get an increase in the rate near threshold, and then the cell becomes inhibited, so you get a non-monotonic rate intensity function. But as you go up if you do wideband noise, it turns out those tuberculoventral cells get inhibited by the D stellate cells, and you get an excitatory response that can continue as you go way up in intensity. And these cells are also very sensitive to the spectral structure of sounds. Now, the cochlear nucleus neurons project to the inferior colliculus. Not all of them do. This is just an example of this. This is a light sheet fluorescence microscopy image of an intact cochlear nucleus, not intact, but unsectioned cochlear nucleus that's been retrogradely labeled with wheat germ agglutinin coupled to a fluorophore 647, go back, in the inferior colliculus. So here's the nucleus, here's the injection in the inferior colliculus, covering different parts of the colliculus, and it's mostly in the low frequency region, which is dorsal in the inferior colliculus. And this is a maximal image projection through the entire cochlear nucleus, showing essentially all of the cells that were back labeled from this projection. So here's the dorsal cochlear nucleus, and here's the ventral cochlear nucleus. And you can see the fusiform or pyramidal cells scattered throughout the dorsal cochlear nucleus. If we take a section along this axis, sort of coronal, and look at it this way, you can see the line of the fusiform cells and these deep cells, which are mostly giant cells in the nucleus. And if you take a cut through what we call the transstrial plane, orthogonal to the acoustic stria, this way it's kind of a horizontal cut, but it's at an angle. You can see the fusiform cells back labeled and giant cells in the deep part of the DCN. In the ventral cochlear nucleus, these would correspond to the population of T stellate cells that are projecting to the inferior colliculus. And remember that the bushy cells don't do that projection, so this is a pretty extensive population of stellate cells. Okay, so the first section here, a little bit about serendipity and the intrinsic discharge of pyramidal cells. So the story here starts when I was actually giving a lecture when I was at Hopkins in a course called Structure and Function of the Auditory System. And I was tasked with presenting this data from this paper from Bill Rohde, Phil Smith, and Donato Ortel, this classic pair from one of the classic pairs of papers that they published in 1983, where they did intracellular recordings from the dorsal cochlear nucleus and the other paper, the ventral cochlear nucleus of the cat, and filled the cells with horseradish peroxidase in order to try to get the structure-function relationship, nail down the structure-function relationships between the discharge patterns of the cells and the types of cells that were present in the nucleus. And in this, they used a paradigm that was fairly common at the time of short tone bursts, 25 millisecond tone bursts separated by 100 milliseconds, and just repeated tone bursts. And when they do this near the CF of the cell, the response to the first tone burst shows a nice regular firing of 5 spikes. Then the cell hyperpolarizes. In response to the second tone burst, you can see there's actually a delay before the first spike occurs, and there's only 4 spikes here. Then the cell hyperpolarizes. In response to the next tone burst, only 100 milliseconds later, there's this little plateau response before the first spike occurs, and that continues throughout the rest of the stimulus. And if you were to take and average all of these together and look at the spike times, you would conclude that this cell was giving you a buildup response, because only the very first one showed an early spike. Everything else showed delayed spikes in response to the stimulus. And as I was presenting this, I was thinking, well, what's the mechanism behind this delay? And there's 2 possibilities. One is that there's inhibition here. And there most certainly would be inhibition, because we know there's inhibitory circuits that get activated. But the other one, which was based actually on me reading of some papers about invertebrate neurons, was the presence of a channel called the A channel, a kind of potassium conductance that activates and then inactivates. And it had been characterized by John Connors, McCowan, and Chuck Stevens back in the 70s. So thought, well, if this is an A current, this could be an intrinsic property of these cells. That is, they might have this discharge pattern because the way their ion channels are set up, that's their preference to have this discharge pattern. And it might, what am I losing here? Okay. And it might depend upon the presence of hyperpolarization prior to this depolarization that's produced by the auditory nerve input. And lo and behold, when we went in this case in adult guinea pig slices with sharp electrodes, somewhat difficult recordings, starting really just a couple of weeks after looking at this, we noticed that when we do an current injection, the cell can fire a regular train of action potentials. If we hyperpolarize the cell a little bit, we can get an onset spike with a long delay between the first spike and the rest of the spikes. So this is very much like the pauser pattern. And if you use either a weaker depolarization or less hyperpolarization, you can also get a delay to the first spike. And that delay, although here it's fairly short, this is about 50 milliseconds. These are on a different time scale. It's about 50 milliseconds. It can easily be up to a couple hundred milliseconds if you set up the conditions just right. And so we wanted to follow up on this and try to figure out exactly what the mechanism is. This is certainly consistent with the presence of a transient potassium conductance, but these cells are big and they're very difficult to voltage clamp. And this was right at the beginning of the time when being able to do patch clamp recording in slices was feasible. And so we moved into doing that. And this is work done with Patrick Knold, where we reproduced this in young rats, which at the time was the only preparation that we could use. And we found that if you change the amount of hyperpolarization prior to a depolarization, we could indeed cause the transition from a regular firing to a delayed firing response in these cells. And the delay depended upon the amount of hyperpolarization that we gave. Interestingly, the first interspike interval doesn't really change. The interval between the first 2 spikes after this doesn't really change as a function of the prepulse. So whatever's producing this is shut off by the first spike. Now, these cells are large, and it's almost impossible to voltage clamp the membrane. You certainly can't voltage clamp the dendrites. It's very difficult to voltage clamp the membrane of the cell adequately in order to study the ion channels. So here we go. We decided to take a different tack, and we pulled outside-out patches off of these cells. So we're using patch electrodes now. We seal onto the cell. We pull back very slowly. We go to whole cell mode. We have access to the cell. We characterize the responses, and then we pull back very slowly. And then you get this little string of membrane, which then seals up, and you have a patch of membrane in which the outside leaflet faces the bath solution. The inside leaflet faces the pipette. It's called an outside-out patch. And when we did that, in most cases, we found that if we hyperpolarize the membrane, this is in voltage clamp now, and then gave steps to depolarize the membrane, that there was a fast, rapidly activating transient potassium conductance, rapidly inactivated, that was present in these patches. We made some measurements of using sort of the traditional Hodgkin-Huxley type of paradigm of how you characterize these channels, looking at the inactivation and the activation. The activation, which we could measure from the currents as a function of voltage at the beginning of the pulse here, shows this rise. It activates a little bit above the resting potential very weakly and then continues to increase. The currents will increase. The conductance saturates here around 0 millivolts. The inactivation, we could measure during this step from this point to this point between minus 112 and about minus 20 millivolts or minus 30 millivolts, in which the current was largest for the most hyperpolarizing step and became smaller with increasing steps and normalized. That's plotted here as the H parameter or the inactivation parameter, and that was best fit by a double Boltzmann. And it turns out there's actually two currents in here. There's a rapidly inactivating fast current, and there's a slowly inactivating current. We then constructed a model which could replicate the responses of the cells very nicely, including this little hump and sag and the different delays to the first spike. And it also could produce the pauser type response. So this response is in part driven by the intrinsic conductances of the cells. I'm going to skip this slide in the interest of time. So just to summarize this, following the hyperpolarization, weak depolarization results in a long latency to the first spike. Strong depolarization after hyperpolarization can produce short latency, well-timed spikes with a long first interspike interval. That's the thing I just skipped. I'm sorry, that's the next one. The intrinsic firing pattern has been seen in guinea pigs, rats, and gerbils, but interestingly, it's rarely seen in mouse pyramidal cells. It's actually it's difficult to evoke it in mouse pyramidal cells. And finally, a brief hyperpolarization of the membrane in the presence of a noisy input signal can increase the synchrony of subsequent spikes. That's the one that I skipped over. But it does say that this conductance or the de-inactivation of this conductance, making it available, helps with the synchrony of firing of cells subsequently. Okay, hearing loss. So one of the things that we've looked at over the years has been what changes in the cochlear nucleus with hearing loss. One of our first forays into this was work that was done by Howard Francis when he was a neurootology fellow at Hopkins. We did a very simple paradigm of using Sprague Dawley rats. We did bilateral cochlear removals at 4 weeks of age, and 2 weeks later we made recordings from the cells in the ventral cochlear nucleus with sharp electrodes, and we saw in the normal rats the normal firing patterns, regular firing patterns for the T stellate cells, and for the bushy cells, single spikes and sometimes pairs of spikes or 2 spikes here. This is the normal pattern that you would see. 2 weeks after deafferentation, complete cochlear destruction, the patterns were basically unchanged. In fact, nothing really changed in the cells except for a slight change in input resistance in the T stellate cells. This is good for doing things like cochlear implants because now you know that the system that you're stimulating has a set of electrical properties that are very similar to what's seen normally in the intact. They're not changing wildly. But it was a little disappointing that we kind of expected to see some changes in the excitability of these cells. There are changes in the synaptic responses of these cells, which we and others have seen over the years, even in response to modest acoustic deprivation. So even though the intrinsic excitability stays pretty intact, the synapses, the end bulbs of Held in particular, are somewhat plastic and sensitive to the acoustic environment and changes in the acoustic environment. It's been shown by Matthew Zhu Friedman, Lonnie Rubio, and Amanda Lauer, and others. So the next set of experiments I'm going to talk about use a slightly different experimental paradigm. And these sets of data are unpublished data at this point. But we used mice. We did a 2-hour noise exposure, 8 to 16 kilohertz octave band, a very standard sort of exposure system, 106 or 115 dB SPL, meant to cause a partial hearing loss or a more complete hearing loss. And then we allowed a 2-week recovery. So the noise exposure was done at 6 weeks of age in these mice, and the recordings were done about 8 weeks of age. This shows a light sheet image of the cochlea, again from Ken Hudson, stained for myosin 7 to show the hair cells, and stained for TUJ1 in the background here to see the spiral ganglion cells. And you can see this is a noise-exposed mouse. And here in the very high frequency region, there's a loss of hair cells starting at this point. Surprisingly, in the region where the noise exposure is most prominent, the hair cells are, at least with this rendition, are still intact. However, the animals have increased thresholds. So this is the sham. Thresholds are around 30 dB SPL. After 2 weeks after 106 dB SPL exposure, the thresholds are raised 50 to 55 dB. And after the 115 dBL exposure, it can be difficult to observe a response, although occasionally that's the case. So this just shows the input-output functions. The amplitude of the N1P1, this initial wave here, is decreased as a function of the intensity of the noise exposure, and the thresholds generally increase. So the first set of experiments, these were done blinded using this paradigm, and they also included a 3-day recovery. So the way the blinding was done was that Tessa Ropp in the lab did the noise exposures, and she did the ABRs, and Mike Kasten did all of the physiology and didn't know what the treatment conditions were. And then the analysis was all done just through a computer program. So this is a pyramidal cell in the DCN, and showing the usual discharge pattern. This is in response to a very long stimulus. From these, we calculated the current versus firing rate curves, and you can see that there's very little change in current versus firing rate for all of these different noise exposure conditions and the different recovery times. There's no change in the adaptation in the pyramidal cells or in the tuberculoventral neurons, which were also sampled in this particular project. There's really no change in the maximum, what we call the hill slope. So we fit these with a hill function and looked at the maximum slope. That's the change in firing rate as a function of the current injection. There was no change in that. There is maybe a small change here in the tuberculoventral neurons, but it wasn't significant. The maximum, the current at which the slope was maximum, however, generally decreased in the pyramidal cells. It's not statistically significant because there's some points, outlier points here, but you can see the same pattern in the tuberculoventral neurons, where with both of these intense noise exposures after 2 weeks, the current at which the slope is maximum is much smaller. So they're left shifted in their firing current relationship slightly. But there's no change in the maximal firing rate. Interestingly, in the pyramidal cells, the spontaneous EPSCs showed a sensitivity to the noise exposure conditions. So there's a big group effect here between the controls and all of the other noise exposure conditions. And the spontaneous rate is increased in both the 104 here and the 115 dB SPL groups compared to the control groups. The 3-day recovery group is kind of all over the place. And there's a change maybe in the tuberculoventral cells, but this was a small data sample, and so there's no significant changes in here. So there are some changes in excitability that occur in terms of the sensitivity to current injection in these cells. Now, what about the interneurons? To look at the interneurons, it's a little difficult to sample large number of interneurons in the material that we were using for those experiments. They were a mouse in which the type 1C auditory nerve fibers had happened to be labeled, and I'm not presenting that data. So we obtained the GlyT2 reporter mouse from Larry Trussell, which labels the cartwheel cells and tuberculoventral cells in the DCN, and then recorded from these cells. So this is a characteristic response of the cartwheel cells, showing their complex spike burst. And these are tuberculoventral cells showing regular firing. This is a diagram from Zhang and Ortel showing tuberculoventral cell in the mouse DCN, its projection to the VCN. And surprisingly, we saw changes in the action potential shapes in cartwheel cells. So we measured the very first action potential in these bursts, and we found that the rising rate of that action potential decreased after noise exposure. The falling rate of the action potential also decreased. So the action potentials became a little wider, and that's measured here in the action potential half width, particularly in the 115 dB group. In addition, the threshold, the voltage at which the action potential initiated, as measured as an increase in 20 millivolts per millisecond here, was decreased. So the cells are a little bit more sensitive to synaptic input. Their action potentials are a little wider. Tuberculoventral cells didn't show a change in the action potential rising and falling rates, although the half width did slightly increase. And interestingly, the thresholds also decreased with the most intense noise exposure condition. So there are also changes in the excitability of the interneurons in the dorsal cochlear nucleus with this noise exposure paradigm. Finally, we looked at the axon initial segment in these cells. We were motivated by the studies from Kuba and Omori's group, showing in the chicken that there were changes in the length of the axon initial segment after sound deprivation in chickens. So we stained for a variety of different markers, but most notably for the ankyrin G, which labels the axon initial segment, which you can see here in panel B4 as these little long lines. We stained for, in this case, for the VGAT mouse, which was suboptimal, and osteopontin 1, which labels the pyramidal cells and some of the cartwheel cells. But it was better to do this in the GlyT2 mouse, where we could also label the tuberculoventral cells. And when we measured the length, or when Reggie Edwards measured the length of the axon initial segment, he saw a significant increase in the length of the axon initial segment in the cartwheel cells. So this is consistent with some changes in the excitability of the cells. And although we don't have the 106 dB data yet, a decrease in the length of the axon initial segment in the tuberculoventral cells. So accompanying the changes in the electrical excitability are some morphological changes in the cells in the region of the cell that actually is involved in initiating action potentials. Okay. I think I can get through this in time here. So the last part of this, I want to talk about the perspective from the cortex. So how does the cochlear nucleus contribute to auditory cortical processing? So looking at it from the other end of the system. And this is work that was done in conjunction with Hiroyuki, Michelle Lee Garcia, Amber Klein were really critical in this work. And we have been participating with Hiroyuki on this as we look at some things that I won't be talking about, both in the cortex and the cochlear nucleus. But I wanted to give you a little bit of a view of the pathway and what contributes to a particular a phenomenon that was observed in the cortex. So normally we think of the auditory cortex as receiving inputs from the auditory system as having a lemniscal pathway, which goes from the cochlear nucleus to the central nucleus of the inferior colliculus, to the ventral division of the medial geniculate, and into the primary auditory cortical areas, A1, A2, and the anterior auditory field. That's a lemniscal pathway. It maintains tonotopy and it's fast. And then there's a non-lemniscal pathway, which has modulatory roles. We usually think of it as being a multimodal association pathway, and it's slower, and it goes into the shell regions of the colliculus, the medial and dorsal divisions of the medial geniculate, and then up to cortex, but not into the canonical layer 4 thalamocortical recipient regions. Amber, in her dissertation project, was doing recordings from the auditory cortex using this approach, doing intrinsic imaging through the skull to first characterize where the auditory areas were, so you can get the tonotopic map and identify where the areas are, then inserting a 64-channel linear probe into A1 or A2, and measuring the current source density in response to clicks in A1 or A2, or measuring multi-unit and single unit spiking activity. And what's a little surprising here, if you look at the source sink distribution, so the current source density is calculated from field potentials, and it tells you when current is entering or leaving the extracellular volume. And an inward current or a negative current is a current sink, is usually associated with excitatory synapses. Sources are associated with the return currents. And in some cases, they can be associated with inhibition. And if you look at the distribution here in layer 4, there, of course, is a very large current sink. This is the canonical input to layer 4 in cortex. But interestingly, down here in layer 6, there's actually a shorter latency input that occurs and produces a sink that precedes the layer 4 input by a few milliseconds. In A2, this was even more pronounced. Layer 4 input is delayed here. That's starting around 15 milliseconds or so, whereas the layer 6 input is starting somewhere around 7 or 8 milliseconds, peaking around 10 milliseconds. And this is a little bit surprising, although if you look in the literature, especially in A1, you'll see reports of this layer 6 short latency input into the deep cortical regions, and this has also been seen in somatosensory cortex. The same thing is seen if you look at single unit or multi-unit data. The latency matches up with the current source density latencies and occurs earlier. So where is this coming from? So I'm not going to go through the entire series of experiments here, but to get at the pathway that it's going to, one of the key experiments was to do disynaptic tracing to A2, layer 6, using a rabies virus. And this was done by injecting the AAV retrocree into layer 6. This retrogradely labels cells in the geniculate that are projecting to layer 6, and then a second injection is made into the geniculate, which will recombine with these, and then retrogradely label the input cells that are then connected to this layer 6 input. There, okay. Now, this summarizes what happens when you do this experiment in A1 or in A2 with the injections in layer 6. Not surprisingly, you don't see much in the ventral division of the medial geniculate, so not much spillover into layer 4, but you do see neurons in the medial division, particularly caudally here in the region near the brachium of the inferior colliculus. And if you look in the inferior colliculus, which are the input cells to these parts of the geniculate, you see in the external cortex of the colliculus, as well as in the dorsal cortex of the colliculus, that there are cells that are providing input to this region, whereas the central region of the inferior colliculus is much less labeled. And this is summarized quantitatively here for a different set of experiments. In both cases, for A1 and A2, the layer 6 input ultimately seems to come through the external cortex of the inferior colliculus. So these neurons are innervated by the shell portion of the inferior colliculus. If you do unit recordings in these areas, which were also done in Hiroyuki's lab, you see the central nucleus has a latency of about 5 milliseconds. The dorsal cortex has longer latencies, it's been reported before. The external cortex has a combination of short latencies and long latencies, and the shortest latencies are associated with recordings from the most rostral portion of the ECIC as compared to the caudal portion, and that's summarized in this graph, and the label is missing. This should say latency there, but the rostral ECIC shows these short latency responses. So short latencies into the medial geniculate, short latencies to layer 6. Finally, experiments were done to look at where in the cochlear nucleus was projecting to this external region. Were there any direct connections? And so tracers were injected, either wheat germ agglutinin or a cholera toxin B. And then the source was looked at in the cochlear nucleus. If you do a central injection in the inferior colliculus, much like what I showed you earlier with the wheat germ agglutinin, this is in a high frequency region. You get cells in the dorsal cochlear nucleus in the high frequency regions, and cells in the anteroventral cochlear nucleus in the high frequency region. DCIC injections here, you also get dorsal cochlear nucleus neurons, combination of pyramidal cells and some giant cells, occasional cells in the PVCN, but nothing from the anteroventral cochlear nucleus. Caudal ECIC injection and rostral ECIC injections here, You get cells from the dorsal cochlear nucleus, mostly fusiform cells and a few giant cells. There are cells around the margins of the PVCN, which you can see in these two figures, and in the margins here in the AVCN, around the edges of the AVCN, a little bit in the core region here, but mostly from the DCN. So the pathway to this fast pathway seems to originate so from the cochlear nucleus, creating a quote unquote, which of course projects into the central nucleus in the rostral MGV. But there's a second pathway, which we can call, which is called the non-meniscal pathway here, which goes into the external cortex of the inferior colliculus, and an A2 goes through the medial part of the medial geniculate, the rostral part of the brachium of the inferior colliculus, and goes into layer 6 and provides a very fast input to A2 as well as into A1. And then there are slower pathways that come through the dorsal cortex of the inferior colliculus and some of the ECIC. So the cochlear nucleus is providing two different sets of inputs here that are acoustically responsive in the cortex. So the DCN as a source of the short latency pathway requires a little bit of discussion here. The shortest latency pathway to the central nucleus of the inferior colliculus from the DCN has been studied by McInturf et al. This is from Dan Lee and Chris Brown's work. They're interested in auditory brainstem implants, but of course they're directly stimulating parts of the cochlear nucleus. The DCN with direct stimulation has a latency in the central nucleus of the inferior colliculus of about 3 milliseconds. The VCN has a mean latency of about 8 or 9 milliseconds, and that's illustrated here. And with the lowest stimulus level, almost all the recorded cells had fairly short latencies. It's only at the higher levels that you see longer latencies. T-stellate cell multipolar axons are thin, so the T-stellate cells are actually not as likely to produce a short latency response as to contribute to the long latency response. And that's consistent with what happens with VCN stimulation, where you almost see exclusively long latency responses in the inferior colliculus. DCN pyramidal cells also respond to clicks, and they have a fairly short latency. There's two studies that have looked at this, Phil Joris and Phil Smith, recording the dorsal acoustic stria, and Don Godfrey et al. In their original study in '75. And the short latency pathway to layer 6 could rapidly activate cortical output layers, because layer 6 is a major projection pathway from the cortex, and or modulate the function of the superficial layers, because there are local connections from layer 6 to more superficial layers. In addition, it could serve as an early alerting mechanism, maybe provide priming cortex for subsequent detailed processing. So the DCN may be involved not just in things that we've been thinking about in terms of spectral processing for example, sound localization in the elevation plane, but for as a pathway that provides an alerting mechanism, perhaps for some kinds of wideband sounds, such as clicks, that be transmitted up to cortex for further analysis. Okay, so I have a few closing comments. I don't really have any time, but just briefly, biology is hard. So these are just some thoughts that I had that I wanted to kind of give to the younger people. Biology is hard. Things are nonlinear, they're non-intuitive. To solve problems, you need to understand the underlying biology. You can't just solve problems in medicine by going in and trying to mess around with the system without understanding the underlying biology. And to understand that, you need quantitative models and hypotheses, which is why we sometimes do modeling to try to understand and predict what's going to happen. Advances in science are in part driven by serendipity, so keep your eyes open, your mind open. Not all research findings start or end with a planned hypothesis. Question everything. Paradigms need to be questioned. Knowledge and understanding are not static. And finally, I think this is really important these days. We as scientists don't really reach out enough to the public to let them know the value of good science and basic research adds to their lives. So we need to stand up for science, make the work that you're doing known, or even in a very general sense, even if you're doing something very specific, let people know what is important for science that comes from science in their lives. And then these are the people that are involved in the work. Mike Kasten, who's here, is with the lab. Reggie Edwards, Ken Hudson is over here presenting a poster at ARO. an undergraduate, Malcolm Lutz, the crew that did the noise-induced hearing loss studies. Thanks to Larry Trussell for the GlyT2 mice. I talked about work from some of these other people, some of whom are shown in this here. And then the collaborators, George Spiru, who I've collaborated with for a long time. I didn't present any of our work here. And Hiroyuki's lab, which is shown here, Hiroyuki, Michelle Lee Garcia, and I guess Amber's not in this picture, but anyway, Haley Acosta, Hiroyuki Sukano, who's been a very important part of the auditory cortex study. And with that, I'm done.
Okay, thank you. It was a wonderful talk. Okay, please unmute yourself and ask question directly. First of all, I see one question from the chat. Megan Wood asked, what was the strain of the mice in the noise exposure experiments?
Excellent. So it's an excellent question, Megan. So we used two different strains. The first set of experiments that I talked about were done in it's called the NF107 mouse that was crossed with the Allen Institute AI32 channel rhodopsin EYFP reporter mouse. The NF107 mouse is a back transgenic that was generated by the GenSat consortium back in the early 2000s. So Nat Heinz and Mary Beth Hatton generated those mice. It's a collagen glycosyl transferase type 2 reporter. The entire promoter sequence. So, and let me back up. The mouse was generated on an FVB background. There's a few other bits and pieces of CD1 and some other things in there, but it's mostly an FVB background. And it's only expressed in the males. And they have good hearing. We've compared them to FVBs as well as CBAs, and they have good hearing. They're a very robust strain. I didn't talk about this, but the collagen glycosyl transferase type 2 from the has been associated during development with the type 1C auditory nerve fibers. So it's not expressed until the type 1Cs branch off for the 1A and Bs. And we've confirmed this to some extent because the cells in the spiral ganglion cells that are expressing this transgene are PAU4 F1 positive and they're calretinin negative. So, so it's a little bit of an unusual line. It's a little bit of a mixed line. We also used it we crossed the GlyT2 mice with it because we wanted both comparable, a little bit of comparable background genetics, but also to cross out the cadherin 23 that's issue that's present in the mice from the C57 black 6 background. We're not quite at the age where that becomes a really important factor, but it could be something that interferes. And we also, in those studies, did optogenetic experiments looking at the type 1C inputs to all the neurons in the cochlear nucleus, which I didn't talk about.
Thank you so much. I figured once I saw what the strains were later, but thank you for answering that so in depth.
Sure.
And great talk.
Thanks.
Okay, we have another question from Sabina Nowakoska. So thank you for the talk. You showed that after destruction of the cochlea, there was no change in the physiological properties of cells. Was it true along the whole tonotopic axis? Pasek and Rubel 1989 showed that after damaging cochlea, the size gradient along the tonotopic axis was gone. Can you comment on that?
Yeah. So we didn't explicitly look along the tonotopic axis. So we averaged across everything. These were, they're slightly older animals. I'm just trying to think. So we averaged across the entire tonotopic axis. These in the rats here, and these were cochlear removals. So there was not a specific region that was left intact. These were actually physical. The cochlea was drilled down until the stump of the nerve root was visible as it went into the brainstem, and that was it. So these animals were quite deaf and actually lacked spiral ganglion cells. We didn't do any anatomical studies on the cells to determine whether or not there was a change in the sizes. So we don't know.
Thank you. Yeah,
Thank
I
you.
was just wondering, I don't remember in that paper, I think they said they did cochlear ablation. I don't remember exactly what was the method.
Yeah.
But I was just, yeah, it's, I find it interesting, like that it was actually on quite a short temporal scale that they then observed changes in the cell size in the
Right.
cochlear use. Namely the loss of the gradient, which means that the low frequency tuned neurons became smaller. So I was just like wondering if that would also somehow be then reflected in physiology. But
Yeah. No, we didn't see anything. They also did that in gerbils, if I remember right.
yes, but in gerbils that was in that was in gerbils, yeah.
Yeah. So they have better.
Also the MNTB and yeah.
Yeah. So they have a more extended low frequency hearing range than rats do also. So it's less represented in that preparation. But no, we didn't. Unfortunately, we didn't look at things. Experiments, there were sharp electrode experiments in adult cochlear nucleus were not trivial at the time.
Yeah, thank you.
Sure.
So I guess I have a question about the lemniscal and non-lemniscal input. So can you tell a little bit more about the cell type, and also how is that? I mean, during hearing loss, which pathways like more impacted?
Yeah, so that's a great question. We don't know which pathway would be more impacted. I mean, from the standpoint of auditory cortex. Can you rephrase the beginning of it with the lemniscal and non-lemniscal input?
You showed some of the tracing data. I wonder about, do you have more information about the cell types that providing those inputs a little bit more?
Right. So we've done recordings. So there are two parts to the answer to that. We've done a lot of recordings in the dorsal cochlear nucleus, and the cells that are back labeled from the inferior colliculus with those injections are the fusiform pyramidal cells and the giant cells. No other cells that we've recorded from yet, or we've had no other cell classes that we recorded from yet. We tried cartwheel cells, and they, you wouldn't expect them to project out. In the ventral cochlear nucleus, it's a little more complex because these are cells along the margin and they're fairly sparse. The few that we've seen seem to be a kind of a small stellate cell. They fire regularly. We're still working on that though. So we don't really know.
Margin, you mean still within the small cell?
Along the small cell cap, along the medial margins and lateral margins of the ventral cochlear nucleus. They're not in the core regions where people are usually recording from. So
yeah, I remember there's some weird neurons there.
there are, yes.
So okay, any more questions? Please unmute or put your question in the chat.
Hi, Paul. Could I ask a question?
Sure. Hi, Adrian.
Thanks for a lovely talk. Really clear. What do you make of this difference between the inputs going to the caudal and to the rostral external cortex?
In terms of the distribution?
Yeah, because there's a difference in the timing of the inputs to those. Is that right? Did I catch that correctly?
Yeah, there's a slight difference in the timing of them. The rostral inputs are slightly faster than, have slightly shorter latencies than the caudal. I mean, there's a little bit there's overlap between them, definitely.
Mm-hmm.
Beyond that, I don't really have any great idea. I mean, there's the projections from the cochlear nucleus look fairly similar. Maybe the caudal region gets, I don't know, maybe there's a bias in the inputs from other regions, and the other possibilities, for example, would be that I was just trying to think about this. I don't really have a good answer for your question. There seems to
Mhm.
be a difference. I'm not quite sure what it is. The rostral ECIC and the caudal portion of the brachium of the inferior colliculus are kind of, we think that they're kind of continuous in some respects. And so there may be something different about the way that the nucleus is projecting up into that area and which cell types. One of the things that I don't have a good handle on is the diameters of the axons of, say, the fusiform cells.
Mhm.
We have measurements, or there are measurements in the literature from Dan Lee and Chris Brown of about 1.4 microns. I think that's the ARO paper for the T stellate cells, but there doesn't seem to be anything comparable for measurements of the DCN pyramidal cells. And so maybe there's a difference in the projections, let's say, of the giant cells, which might have larger diameter axons compared to the fusiform cells. But the data doesn't really support yet a difference in the projections to those different to the caudal and rostral regions.
Okay. Yeah. Great. Okay. Thanks.
Yeah.
Thank you for your thoughts on that.
Hi.
Hi.
Thank you for the fantastic talk. Have you considered the direct projection from the dorsal cochlear nucleus to the medial division of the medial geniculate body as another source of this short latency pathway?
Yes. Yeah. So I didn't show the data in the paper, which is in press. We make a comment that that projection doesn't seem to be as large from the dorsal cochlear nucleus. It's not as large as the projection to the external nucleus of the inferior colliculus. So it's present, but it seems less likely that it actually contributes to this short latency pathway.
I see.
Compared to the weight of the projections from across the pathway that I described. So
well,
it was one of the early contenders.
Yeah.
Before some of the experiments were done. Yeah.
Thank you very much.
Okay, thank you.
Okay. If there's no more questions, thank you, Paul, for the wonderful talk. And we'll also thank all the audience. Please pay attention to the ARO email. We'll have more talks on this series. Yeah, thank you all.
And thank you.